I'm Sarah Fitzsimmons. I'm the Director of Restoration for the American Chestnut Foundation, and I work out of Penn State. We've had a couple of requests for information about transgenic trees this morning, and a couple of other questions. So both in the Q&A and in the chat box, I think Sharon put a link, thank you Sharon, I think that's you, put a link to our past Chestnut Chat series. So I encourage you to check out that link and check out our past webinars. If you have a question, we encourage you to use the Q&A format because that allows us to keep the questions and the answers together. It's a lot easier for us to track it that way. You can use the chat. You can make comments over there. That's a great place to do that. And if you do ask questions, we'll keep up with things over there, but we encourage you to use the Q&A instead. I'm going to turn things over to Lisa really quick and then take it back over, and we'll go to Hoy and Hill.
I'll be really brief here. Good morning, everyone. Glad you're joining us. These Chestnut Chats have turned out to be really popular and a great way we can all engage as a community while we're sheltering in place. So I'm really happy this morning to introduce the Hoy and Hill show. They're amazing plant biologists, practitioners, and plant whisperers, chestnut whisperers. So I understand that grafting chestnuts is really tricky, and they're going to share some of their trade secrets today and hopefully answer some of your questions out there in the hinterland. So thank you again for joining. Thank you to all of you that have donated to the Spring Appeal. Our theme is thriving, not just surviving, but thriving. So it's really amazing. I'm trying to call all of you and thank you personally since I'm sheltering at home. So just wanted to throw out a thank you there for all of you that are continuing to keep the American Chestnut Foundation strong during this tough time. So anyway, I'll turn it back over to Sarah. Correct? Are you going to introduce Hill and Hoy?
I will.
Very good.
Thanks, Lisa.
Thank you.
Yeah. So really quick, I'll let Hill and Hoy go into a little bit more information on their background, but I've had the pleasure of working with Hoy directly at Penn State since 2014. Is that right, Hoy? Did I get that date right?
Yep.
So Hoy is the orchard manager, the Penn State chestnut orchard manager, and we work together out of that office. And just, I have to tell this anecdote because it cracks me up. The Pennsylvania chapter hired Hoy to help out with the orchard process at Penn State after while I was pregnant with my first child, Parker. And after we hired Hoy, I was having dinner with my in-laws, my husband's parents, and I said, yeah, we just hired Stephen Hoy. And he goes by Hoy, and they're like, really? Hoy? We think we know someone like that. And it turns out, and I didn't know this, that Steve's dad taught my husband canoeing and outdoor skills, and also shop in high school. So it's a pretty small community around State College. But since then, I've had the pleasure. Hoy is amazing at all things mechanical and can fix anything, and grows chestnuts marvelously, and has been doing grafting now for a couple of years and working on perfecting his technique. So Hoy is going to cover a sort of the beginner's perspective on grafting and what it takes to get things going. And then Hill will cover sort of the more seasoned veteran style of grafting or perspective on grafting. We ask, or I'm going to be holding all questions until the end. Again, please use the Q&A for your questions, and I'll be feeding Hill and Hoy the questions after they give the formal presentation part of things. So with that, Hoy, you want to take it away?
Sure. Good morning, everybody. Thank you again for joining us, and hopefully this is a relaxed but informative webinar on grafting. I would say I fall squarely in the novice category when it comes to grafting, specifically chestnuts. And this is the year that I've really taken on any substantial grafting, trying to accomplish 100 plus for the year, and hopefully most of those will be successful. All right, hopefully everyone can see this PowerPoint screen. So one of the biggest questions is, why do we graft? And specifically, this is why am I grafting here at Penn State and the Pennsylvania chapter? So you'll see three examples across the top. We have a pure American chestnut tree. This is in south central PA on the edge of a talus field, which is the home of the Allegheny wood rat, and they really love chestnut. So this is an area where you'll typically find these clusters of sprouts, but you can see that the tree is not in the greatest health, probably not going to continue to survive for many more years. So this is something that it's a difficult site to get to. It takes a long time. This would be a good candidate for trying to harvest scion wood off of and bringing it back to the office for grafting purposes. The middle photo, you see a very suppressed seedling that's in the understory. Again, not an ideal situation for a chestnut tree, and another good candidate for grafting, collecting scion wood off of that, because it's definitely not going to be in a position to produce flowers or nuts in the near future. The third tree on the right hand side is a very mature American chestnut, and this is also in Pennsylvania and north central PA on state game land property. So again, isolated specimen, might produce nuts, but the odds of us being able to get out there and have the appropriate timing and methodology to harvest the nuts is difficult. So if it's producing sprouts, or if we can find, maybe some offspring of this tree, that would also be a great candidate. So we want to bring all of that material back into a more centralized area where we can have the grafted trees. We can hopefully have success in those grafts, and then put them in a germplasm conservation orchard, or a more concentrated area, so we're not taking several hours to drive to these locations to harvest one tree, and then drive several hours to another location for one tree. We can go to one site, harvest many trees, and maximize our time in the field. So again, this being the first year that I really went down the grafting route in earnest, did not have ideal supplies for grafting, especially after a couple conversations with Hill. So at the bottom left-hand corner, you'll see what I was working with. And some of this was based just off of what we've used in the past. Some of this was based off of what we've found visiting some commercial nursery operations and what they were using for their grafting. And some of it was, a good old Google search of grafting fruit trees, grafting trees, necessary supplies. So I'll just walk through a few of these items. So kind of working clockwise from the top left, we have our parafilm, which replaces a grafting tape. This is used to wrap up your graft after you've made the union. Good old Elmer's glue, which is used to cover any of the open cuts, say on the end of your scion wood, or if you're doing a cleft graft, you have a lot of exposed wood on your rootstock, you would use that to try to seal that off. Not ideal, and I have a feeling that Hill's going to go into this in more detail, but some cons to working with a product like Elmer's glue is it's not as elastic as, say, grafting wax would be. Some of this glue gets washed off in the rain, or over time it just kind of deteriorates, so it could be detrimental to the graft in the long term. We have alcohol or some other material to sterilize your tools, sterilize your scion wood. This is ethanol alcohol. but isopropyl can work. A dilute bleach solution can work depending on what you're working with. This is a product which actually has no English on the package, but I believe they are tomato clips for grafting tomato plants. So they're a little spring-loaded clip, and you use that specifically. We're using that with whip and tongue or a bark, a chip bud graft, where you'd use that to just help encourage better contact with your rootstock and the scion. Pair of clippers here in the middle to make some of your general cuts, get your scion wood cut down to more appropriate size. I did not have a grafting knife, and that's something that I will definitely be picking up for next year, but a utility knife, an X-Acto knife, works relatively well. There are some considerations for it. Specifically, they are what's called a double beveled edge. So they're sharpened on both sides to a nice point, which is great for general cutting, but whenever you're dealing with scion woods and rootstock cuts, they tend to want to curve as you make your cut, and it's very difficult to get a nice flat plane so you have solid contact between your rootstock and your scion. So that was something that required a lot of effort and concentration to make sure that you're getting those nice flat cuts or as close as you can, so you have your good contact, and that is going to be essential for the success of your graft. And then lastly, but very important, is some sort of documentation or tagging method. So these are just pot sticks and a permanent marker, something to write information on your graft so that you can label that pot, label your graft and your rootstock, and you can track that down the road, and you know what grafts belong together and what needs to be separated out. So the top center here is a grafting knife. This is a Victorinox brand grafting knife. So this is what I will be trying to pick up for next year. They're single bevel, so only one side of the blade is sharpened, the other side is flat, and that helps to keep your cuts nice and flat without the curving that I mentioned for your X-Acto knife. And then lastly, you have grafting wax here in the upper right, and that's going to just be a better option for folks than Elmer's glue. I also found several places that indicate a toilet wax ring that you use to mount your toilet to the floor. Works very well for grafting. I haven't experienced that. I don't know if Hill can speak to that, but might be a cheaper and more readily available option for folks if you're just making do with what's locally available. All right, so into the actual grafting world. I'll start low and then hopefully end on a high note. Did most of these grafts back in March, and out of those, I think it did close to a 50 initially, and then did another round of 50 later. And a majority of them here in the upper left, this is a cleft graft, or sometimes referred to as a heroic graft. So we're dealing with rootstock that most of these came from trees with poor form, so they weren't going to be ideal for planting out for ceremonial plantings or educational plantings, but they make excellent rootstock. So went ahead and cut off a majority of the crown and put the scion wood here on the side. In this graft, you peel back the bark on the rootstock, and you take your scion wood, which for these is generally smaller than you would want. They're only a couple millimeters in diameter, and they're coming off as suppressed sprouts for the most part. And you're just trying to harvest those genetics and get them into a place to conserve them for later use. So making small cuts and putting that scion wood into the edge of your rootstock, and then you're wrapping it up. Again, putting Elmer's glue on the very top of that rootstock to prevent drying out, putting glue on top of your scion wood to prevent that from drying out. But you can see this graft did not take. It did not even start to leaf out. The scion wood just kind of withered and then died, and you see some epicormic sprouting from your rootstock here. So that's what about 50% of my cleft grafts look like, for a variety of reasons. I think part of it was, as I mentioned, just the lack of good contact between the scion and rootstock. But hopefully Hill can help troubleshoot. The second one on the left is a whip and tongue. Again, dealing with small diameter material, just poor contact, and it started to leaf out, but then ended up withering and failing. So same thing with these two on the right, where you had partial leaf out or no leaf out of the scion wood, and then you get your epicormic sprouting and ultimate failure of your graft. So not what we want to see, but something it seems like most people grafting chestnuts have experienced. On to the success. So this is what we're all hoping for. Again, about 20% of my grafts that survived were cleft graft. The other majority were the whip and tongue. So the cleft graft here, you can see some small leaves. This is on the far left of the screen. So some small leaves leafing out. You still have your epicormic sprouting. So it'll be a waiting game to see how this performs, but initially some good success. This one, second from the left, has a great graft. So it extended about 15 inches so far, has many leaves, and it looks really happy. Little to no epicormic sprouting on the rootstock. And then the whip and tongue, I did these first and then realized that my scion's too long, but still had the terminal bud leaf out in both cases, and they're both doing pretty well. So this is what we're definitely hoping for. Again, squarely a novice in this field and hoping to improve greatly each and every time. But it takes practice. You know, a big hurdle for me was just getting over the intimidation factor of grafting. So take your time. Maybe I found reducing your caffeine intake for that day. That helps a lot to just reduce the shaky hand and help improve your cuts. All right. Thank you very much. That took a little longer than I expected, but hope that was informative, and I'll turn it back over to Sarah.
Great. Thanks so much, Hoy. And like I said, we're going to hold all of our questions until the end. So I'm going to go ahead and turn it over to Dr. Hill Craddock, who is...
Sorry about that. I didn't hear anything.
Is that Siri? Biology at the University of Tennessee at Chattanooga. And Hill, how long have you been doing grafting of chestnuts? Of anything, actually. Let's say anything, and then of chestnuts.
I joined the Northern Nut Growers Association in 1978. So I was still a teenager. Wow. I was 18 years old in 1978, and I bought my first grafting knife right about that time.
Huh.
And I just grafted a couple things, didn't have much success. By the time I was a graduate student in the early 1980s, I ended up in Corvallis, Oregon, working at a USDA ARS lab. It was the National Clonal Germplasm Repository in Corvallis. And that sounds very sciencey. It sounds kind of scary. National Clonal Germplasm Repository. And essentially what the germplasm repository is a gene bank for fruit and nut trees. And so Corvallis, the location at Corvallis has the world's largest collection of pear trees, hazelnut trees, hops, mint, and blueberries, vaccinium, and then raspberries and blackberries. And so part of my job there I had a research assistantship, besides digging irrigation ditch and fixing the plumbing, was grafting. And so I had a very good mentor, guy named Don Byers. And I still remember my first day on the job. I was up on, Don was on the ladder and we were building something. And he said, "Hand me your pocket knife." And I said, "I haven't got a pocket knife." And he came down off the ladder and he looked at me and he said, "If Harry," that was my major professor, "if Harry finds out you showed up for work without a pocket knife, you're in trouble." And so the very next day I got to work, I walked into the lab and Harry says, "Oh, Craddock, come here." He called me into his office. I said, "Good morning." And he said, "What's this I hear about you showing up for work without a pocket knife?" Don had already busted me. And so anyway, Don taught me how to graft in a serious way, and we did bench grafting. I also worked for a plant pathologist. We studied viruses, and many viruses are latent. In other words, they have no symptom in the plant. These are plant viruses. And so we were doing virus indexing, and to do that, we had to graft virus sensitive type. So we had to produce thousands of these virus sensitive types. And then we had to go back and we had to regraft on them a sample of the unknown. And so each plant actually got 4 grafts. One was to graft it to the virus tester. And then the other 3 were little bark grafts that would transfer the virus from the unknown tree to the virus, because viruses can be transmitted by grafting. And so by the end of those couple of years, I had made tens of thousands of grafts. And so that was where it really, so I moved to Italy in 1987 to be married. I found out that was an employable skill I had. And I started working, I started doing piecework for some commercial nurseries and found it very rewarding while I was working on my doctorate at the University of Turin in Torino, Italy. Did I answer your question?
You did. Thank you.
So it's a long time. I've been grafting a long time. So, my experience grafting chestnut trees in particular is mostly from my Italian experience. So I lived in Italy for 7 years, but that was 30 years ago. Like I said, I moved to Italy in 1987. I'm gonna start my PowerPoint now. That's okay with you, Sarah?
It is.
So I'm gonna repeat some of what Hoy said about why we graft. First of all, grafting grafting occurs in nature. I just want to say that plants graft themselves in nature, and it was humans observed this. I'm sure the ancients saw that if 2 plants are growing in close proximity and they touch, the tissues can fuse and they can become one plant. Roots graft, even limbs can graft, and you may have observed that a beech tree is a beautiful example where the limbs, as they grow, they might cross or touch, and then they'll fuse together. Grafting, though, in nature is rather rare, and the observation would have been that like things graft. In other words, a beech tree could graft on itself, but not an oak tree is going to graft on a beech tree or something like that. And so the observations then about grafting come from this ability to, it's a way of fusing plant parts together. And so it's a way that we use to asexually reproduce plants. And so we call these asexual reproduction clonal propagation. And so it's a very, very ancient technique. We have evidence from ancient writings from China and from Persia and from ancient Greece, the classical Greeks. knew about grafting, and they were grafting olives and chestnut trees. In fact, some of the oldest grafted plants in the world are olive trees that still exist in Sicily and parts of Greece and the Mediterranean. So it's a very, very old technique for reproducing a superior genotype. For instance, wild olives are pretty much worthless. They're inedible. They don't have much oil. They're really tiny seeds. But somebody discovered a mutant on the mountain over there that made an edible olive that had a lot of oil in it. Olives aren't really edible. I shouldn't have used that as an example. They had a lot of oil in it. And they wanted to grow that kind. If you grew the seeds, they come up wild. So the only way to reproduce that superior genotype was through an asexual or a clonal method. And so grafting became widespread then in the classical world to propagate these superior genotypes. Now, it's not the only way. If you could grow a cutting, a grapevine, you could just root a cutting and it would grow in its own roots. But an olive branch or a chestnut, very difficult to produce roots. They just don't like to root. So what we do is we grow the roots from a seed. So if I'm going to graft a chestnut tree, I need some roots, and I'm going to harvest my chestnut seeds, plant them, and I'm going to use those as the root stock. And so then the top I put on it is the cultivar. It could be a commercial cultivar because I'm interested in growing that kind of chestnut. I want to grow the Italian Marrone because they're the best chestnuts in the world. And so I have to get a twig then, a branch, a cutting from that original tree, which may have been born as a seed 1,000 years ago. I'm going to graft that now onto my root. Just to make sure everyone is clear, the grafted plant then is a dual organism. The roots maintain their own genetic integrity. There's no mixing of genes. There are fluids that move back and forth, the sap, and there are sugars and hormones that flow up and down the stem. But each part is genetically different. And so the scion maintains its own genetic integrity. Now, there are changes that can occur at the physiological level. For instance, some grafts cause a dwarfing effect of the scion. So for instance, an apple tree, if you grow an apple tree from a seed, it grows into a big thing 50, 60 feet tall. But in some combinations, if you graft that apple type onto a special rootstock, and the rootstocks have been selected for this, and by the way, those rootstocks are grown from cuttings. Not all apples can grow from cuttings, but the best rootstocks can be. You get a dwarf apple tree. And so genetically, it's not changed. It's changed physiologically because of the way the graft union moderates the flow of information and hormones and sugars and water up and down the stem. It could cause a dwarfing effect on the plant. Hoy mentioned one of the reasons he grafts is to capture this wild diversity of naturally occurring American chestnuts that are threatened by diseases. I'm going to add in the South is particularly critical because we have, in addition to the chestnut blight disease, we have the Phytophthora root rot. And worse than blight, Phytophthora kills also the roots. So when they're dead, they're gone. They're not going to resprout from the base. So there's this critical moment where we still have wild surviving trees in nature and that we can collect grafts from them without disturbing the roots, and hopefully we can conserve this germplasm in an ex situ way. And so, I think in terms of conservation, we want to talk about conservation. Of course, we need to conserve forest lands. Forests are the reservoirs of genetic diversity. But it's not always practical, or in some emergency situation, we need to take some extreme measures. And so by creating a germplasm conservation orchard, we might be able to prolong the life of some of these plants, and we can capture their genes in the form of a disease-resistant hybrid. So these grafted trees, the grafted American chestnuts that we're collecting from the wilderness, they don't have to live 100 years. if they live long enough to produce a flower and produce pollen, or even better, a burr, we can capture their genes in the form of a hybrid. And because grafting changes the physiology, it can sometimes shorten the period to blooming. You know, a seedling tree could take 5 or 6 or maybe 10 or 15 years to produce a flower. Grafted trees typically bloom right away. 1 or 2 years after grafting, they can produce flowers. And so it's a very good way then to accelerate the breeding program. We can graft scions from trees that are growing in the woods, or from trees that have never flowered because they're growing in the shade. We can bring them into a nursery or an orchard, or even into a light chamber and shine a bright light on them, and we can force them to bloom at a very early age. You know, within a year of grafting, we can have flowers on trees. Another reason for cloning the chestnut, of course, is to study at the genetic level. Hoy mentioned about how many miles and how many hours it takes to get to the trees and back. By having a germplasm collection all in one place, you can sample all of that diversity in a single location. And that can be very helpful for the researcher who's interested in studying genetic diversity. Also for studying the interaction of genes and the environment, studying the interaction of the host and the pathogen. You may need to have multiple individuals, because if the sample technique, you know, if you're going to give it blight and watch it die, you've killed it. How do you have a control? How do you replicate that experiment? Well, you have to have clones. If you want to test that genotype, you have to clone the plant first. So I've got 10 copies of the same individual, and now I can inoculate some, and I can not inoculate the other ones, or I can inoculate them and study them over a time course. But if the sampling is destructive, I need to have multiple copies of the individual. So we can't root cuttings. It's very difficult to get mature trees into and out of tissue culture. And so grafting turns out to be a very practical method of propagating for all of these reasons. Just wanted to go through a couple of slides. I can't see you, so I can't see your body language. And we're going to wait till the end for questions, apparently. But these are some photographs of some grafted American chestnuts in Italy. And I don't know if I wiggle the cursor here, I don't know if you can see, but that's the graft union. This tree was grafted probably more than 100 years ago, maybe 150, 200 years ago. This is a European chestnut in northern Italy. You can clearly see the blight has affected these trees, but they've been pruned. They've been inoculated with hypovirulent strains of the blight, which is the attenuated form, the virus-infected form of the blight. And so they're recovering from chestnut blight, and they're being maintained as fruit-bearing trees, even though at this very great age. The point of this is that these represent a clonal forest, a forest of grafted trees, very, very old. In fact, this is a monumental tree. It has a tag on it from the Italian Forestry Division. It was grafted probably about head height, because that's a little bit higher than a goat will browse. And so you have to think about that. If you're grafting, you want to put your graft higher than goat browse. There are all kinds of grafts. Tom Scielli, some of you know Tom, asked me in particular to show some pictures of in-arch grafting and bridge grafting. And these are photographs I took in Italy. The 3 smaller photos are bridge grafts that have been used effectively. Now these were young, and so they hadn't grown out very long. And the one on the right is an in-arch graft. Now this is a technique that would bridge or cover or help repair a tree after some damage or wound. In this case, it was chestnut blight. And so the trees had a canker that was removed, a virulent canker. They were treated then with the hypovirulent strains, which works very effectively as a biocontrol on European chestnut and on some of the hybrid Japanese European hybrids. And so the tree on the right, then you can see that a root sprout, a sucker from the rootstock was grafted back in to the scion, and forms now what's called an in-arch. Now this is not a new technique. Arthur Graves wrote about this. In fact, there was some circulation, there was some chatter on the internet recently about some publications of Arthur Graves from the 1950s, where he had pictures of in-arch grafts that they were doing to keep their breeding stock alive at the Connecticut Agriculture Experiment Station and at his home in Hamden in Connecticut. The graft on the left is called a whistle graft. And this has to be done in the timing is just right when the bark slips, and in the growing season when the sap starts to rise, there's a moment when the bark slips, and you can actually remove the rind off of the stalk. And so you take a little piece of this scion when the bark is slipping, and you slip it over the end, so it's a hollow piece of xylem, a hollow piece of phloem tissue, and you just slip it over the tip of the stalk, and it grows. The graft on the right is what I call a heroic graft. This is typical of scion wood that we get. This is not ideal scion wood. I'm showing this as an example of don't do this to me. Don't send me scion wood where the last year's annual growth, I don't know if you can see where I'm jiggling the mouse out here at the tip, but that tree made about 1 millimeter of new growth. In other words, not much vigor. 1 or 2 buds at the tip of each one of these little twigs, so that to get a piece of wood that I could hold on to and cut with my grafting knife, I had to go back 1, 2, 3, 4, 5 years. So this piece of wood here is 5 years old. Now my rootstock is a 1-year-old seedling. And the problem there is that the thickness of the rind, the phloem tissues, are different on a 1-year-old stem and a 5-year-old stem. So the cambium layer is not going to line up right. Okay, so a little bit of vocabulary. The cambium is the vascular cambium is the lateral meristem from which new xylem and phloem cells will be produced. And the xylem and the phloem are the vascular tissues of the plant, which are the vessels that move water and dissolve nutrients up, and then sugars and vitamins down. So the graft union is where we have to reestablish that vascular continuity from the stalk to the scion. And so to get a successful graft, we have to line up those vascular elements. And so to do that, we need to match the cambium layer. So the cambium layer, which is this meristem, meristems are undifferentiated cells. The meristem is going to produce phloem tissue on the outside and new xylem tissue on the inside. And if it matches up, we can reestablish that vascular continuity. Here's a commercial chestnut orchard in northern Italy. Just had a couple of pictures I wanted to show just because I like chestnut trees. Every tree in this photograph, every tree is a grafted tree. These are clonal forests, and they can live for thousands of years. Of course, modern commercial orchards are also based for horticultural purposes. You're going to grow cultivars. This is a chestnut cultivar orchard in Michigan. It's a photograph by Dennis Fulbright, who passed away just last year. You may have known Dennis. We've got examples here in Tennessee. This is one of my students and one of our favorite cultivars here in Tennessee, but all of these commercial cultivars are propagated by grafting. This one's a really good one. The tree is sort of a dwarf, but the nut is full size. It peels really well. It's delicious. It's Chinese chestnut. It's named after Jerry Payne, who was an entomologist from Georgia. I mentioned accelerated breeding. If you can get your trees grafted into an orchard, they'll bloom sooner. It's less dangerous. You don't need tall ladders. You don't need bucket trucks. You've got all your germplasm in one place. It'll bloom. Like I said, they typically bloom even right away. You know, this is a 1-year-old seedling here on the left. It's blooming. I was able to this plant, I collected pollen from yesterday. I took this photograph yesterday in the nursery. Trent Deason, one of my grad students, Trent Deason grafted this one. Here's a photograph from a couple years ago. Probably shouldn't do this for the sake of the plant. Here's the graft. You can see grew one shoot, made a burr, and now the poor thing, it doesn't know which way is up. It's just, probably you should pluck that fruit off and let the tree grow a few years before it produces a nut if you're interested in the tree. If what you really want though is the genes captured in the form of a hybrid, imagine this is a Chinese chestnut. Imagine this was an American chestnut from some far off into the wilderness where you had to risk your life to climb up that mountain to get the scion wood. You brought it back to the nursery. All you want is the genes captured in the form of a hybrid, or that's your mother for your transgenic pollen, right? There they are. There's 2 seeds. I've already made 2 seeds on that plant in 1 year in the container. All right, so how we graft. Knives. Hoy, talking to Hoy now. Get yourself a decent knife, man. The X-Acto blade, I don't know. Okay, you need a good knife. Let me just talk a minute about knives. I've got a couple here, my favorite knives. Today's talk is not the how-to tutorial. I'm going to do that later, I hope. People have been bugging me about it. I'm going to make like a how-to video of grafting, and I'll go over the technique, my technique, and what you'll see, the way I was taught. Every cut I make is towards some part of my body. So maybe you'll use the knife differently. You'll have to ask someone else to teach you. There are right-handed knives and left-handed knives. And the reason there are right-handed knives and left-handed knives, Hoy alluded to it, is because a grafting knife is only beveled on one edge. One edge of the blade is flat. The other edge is beveled. So I only need to sharpen it on that one side. And the reason is, somebody figured this out a long time ago, the ancients, that to make a flat cut, to make a perfectly straight flat cut, a single bevel works better. So I'm going to make matching cuts on the stalk and the scion. And in the photograph here on the right, you can see I've made 3 cuts. I've made 2 whip cuts, which is the first cut through the stalk and through the scion. They're the same length, and the scion and the stalk match in diameter. And then I've made a second cut on the stalk, which is what I'm going to call the tongue. This is a whip and tongue graft. Hoy alluded to that. He mentioned that. I haven't yet cut the tongue on the scion because I'm just checking to see if they look good, right? But what you can see in this picture, I hope, is that the cut surfaces are flat. Okay, the whetstone, and I just brought my little whetstone. Buy a good whetstone. The knife costs 12 bucks. You can buy the knife. You can buy a set of them. The whetstone, a good whetstone might cost more than the knife. It costs 5 times as much. Buy a good whetstone. This one is a synthetic whetstone. It's got a coarse side and a fine side. I basically only use the fine side, and I use water. So this is I don't put oil on my whetstone. This is just water, and that's because I don't want any oily residue on my knife. It's just a water. So I do the wetting under in water. Okay, and I keep it wet when I'm doing that. The tape I like to use, there's a lot of kinds of tape. You can use any material. You can use electrician's tape, that plastic electrician's tape. It comes in lots of colors. You can get red, yellow, blue, green. It's elastic. It's really sticky. It works really well. The problem is it doesn't expand forever. It's not infinitely elastic, and it doesn't biodegrade. It doesn't photodegrade. So eventually you have to cut it off. You have to remove it. Better is a parafilm, and I buy this. This is a parafilm budding tape, and I particularly like this one because it's parafilm. If you haven't played with it, it's actually wax. It's not sticky. It's wax, and it sticks to itself when you stretch it. And it gives me a very tight seal. It's airtight, it's waterproof, and it works really well. That's my favorite. It comes in different widths. It comes in a couple different brands. You can buy the authentic parafilm brand, or you can buy a knockoff brand. Some of it comes with, this one has a little more tooth in it. It's got some fibers that make it a little stronger. It's got some fibers in there. I don't know what, it's not like regular parafilm. It's like a modified parafilm that's made on purpose for grafting. The grafting wax, I've seen glue, I've seen Elmer's glue used before, and Hoy mentioned it. Here's mine. I'm going to put in a plug for Trowbridge's grafting wax. The wax is important because it's waterproof and it's airtight. This is a mixture of beeswax and lanolin, and it melts. It's solid in here. It's solid here, but it melts right around 98 degrees, between 98 degrees and 100 degrees Fahrenheit, which means human body temperature. So you put a little in your hand and it kind of liquefies. If you put it in a pot and bring it to a simmer, you can make it really liquid, but it's really sticky. And because it's waterproof and airtight, this is the material, a grafting wax, that I would use to cover all of the cut surfaces in my grafts. Labels, I'm going to mention labels. Hoy showed pictures of the permanent marker, and this is something that I've learned over the years, that the permanent marker, like the Sharpie brand, right? Who doesn't use Sharpies, right? Is permanent for about one growing season. That's it. You get 1 year. Then it wears off, and then it's gone, and you'll never know what it said on there. In other words, you will lose all of the information. So my advice is to use a graphite pencil. Graphite, it's inert. It lasts forever. You can fossilize it. It'll last 10,000 years, I guarantee. It'll never, doesn't degrade. It's an inert compound. Graphite, okay?
You know, just to jump in here, we want to give you a little time check. We're at 12:20, and I know we all have another talk at 1 o'clock.
I've got 10 minutes, right?
Well, if you can,
It's time for questions.
That leaves time for questions. Yep. So if you can, but I also wanted to say, in defense of the labels that Hoy showed, those are an ink valve, a paint ink valve marker, not a Sharpie.
I'm skeptical. Or use an embossed metal label. Embossing. Emboss it in metal. All right, so here's a picture of my graph kit. On the left, I've got a couple of knives. The plants, that's just a temporary marker I'm going to put in the stake. This is good scionwood. I wanted to talk about good scionwood. I hope you can see that. Good scionwood is from vigorous 1-year-old growth. It should have a bud at every node. The internodes should be long enough that the buds are spaced out so that I can make good cuts. It comes in a variety of diameters, about pencil thickness, maybe a little bit thicker, maybe a little bit thinner. Okay, the rootstocks are going to be 1-year-old seedlings. I'm going to put that graft as low to the ground as conveniently possible because I'm going to get a more vigorous shoot. If I graft way up at the top of tippy top of a flimsy little stem, it's going to be a flimsy little shoot. I want a nice vigorous shoot to come up. I don't want any other part of the rootstock except the roots. I'm going to graft it right as close to ground level as I can. So quick vocabulary: ortet, ramet, genet. These are some terms. The ortet is the original seedling from which we get the kind. Ramet is the propagated plant, the cloned plant that we get. And the term genet is sometimes used to define that whole clonal population. The scionwood, of course, is the part that goes on top. The stalk is the rootstock. Vascular cambium, meristem, xylem, and phloem we talked about already. Some pictures of some grafts. On the left, a whip and tongue. On the right, a chip bud. You see those? A modified whip and tongue. These are things I will go over in my how-to video. You can use rubber bands. I like parafilm better than the rubber bands. It's easier to apply. It doesn't work better, really, but it's easier. And then a whip and tongue after it's been grown. Advantage of the whip and tongue, and I know I'm taking up time. The advantage of the whip and tongue is that the wound is small and it heals quickly. Cryphonectria parasitica is a wound pathogen. And so the sooner that the wound heals, the quicker it is. For scionwood like this is challenging scionwood. This is from a mature bearing tree. It's not vigorous. There are not buds at every node. A lot of these nodes have no buds, and they're very thin. I had to use a different type of graft. This is a bark flap graft. Still as low as I can, you can see the bark flap. Here's another bark flap outside, a cleft, on a top working. And I've got some pictures to show how these grow up. I put 2 on there. This is a bark flap, 1 on each side. And you can see this is the same tree a little bit later. Here's that same crotch, a beautiful crotch. A few years later, we can top work T-buds. We're not going to go into T-buds. Problems that can occur: gall wasp, chestnut blight, grafting compatibility. This is delayed graft failure. Some resources. This will be on the, hopefully, I can send my PowerPoint to Lisa and to Sarah, and she can link to this if you want. There's the best book ever written. It's one of the best how-to books ever written on any subject. There you go. And some sources for knives, and that's my talk.
Thanks, Phil. You flew through that last part there.
Well, I want to leave time for questions.
Yeah, absolutely. So I'm going to give a few questions.
Sharing.
Yeah, I'm going to give a few questions and then hand it over to Hoy at 12.
But first, I'm going to give a shout out to the Tennessee B3F2s there. Look at that.
They're beautiful.
Thanks.
I'm going to ask one of these questions. Ken Law asks, where can I get the long seed pots?
Maybe I don't understand the question, but I'll jump in. The long seed pot, as in a pot that's long?
I think it was one of the, you showed it in one of your pictures. Maybe it was the tree pot, the 1 gallon tree pot, or the.
I buy those from Stewie and Sons, S-T-U-E-W-E, Stewie and Sons they're in Oregon. And you know, the funny thing is they make them in Alabama sometimes, and then they go all the way to Oregon, then they come back to Tennessee when they could have just thrown them here from Alabama. Alabama's right over there.
I've got a couple questions about timing. So Mike Alcott asks, what time is it? When do you best collect scionwood? And then when do you graft?
Can I answer that one, Hoy?
Go ahead, please.
The answer is it depends. And there's probably some grafting you could do year round, depending on the physiology of the stock and the scion, and if you have access to a heated greenhouse and not a heated greenhouse, if you're doing bench grafting, which means sitting at a bench or a table, versus field grafting. The type of grafting that I mostly do uses a dormant scion, which was collected during the winter, but before the polar vortex has frozen the tissues, right? Stored in a refrigerator in a sealed plastic bag with all the air pressed out, not too wet, not too dry. And I've got a stalk that's actively growing. So the stalk should be out of dormancy. It should be actively growing. And so that's the graft I prefer. And so I have a window then, because I have a heated greenhouse, from maybe the end of February, early March through April, May, where I've got a nice window, right? For other kinds of grafting, for instance, I showed a picture of the whistle graft. That's done pretty much in May and June. That's it. You've got May and June. That's all the time you can do. I went really quick. In fact, if I shared my screen, I could go back and show you, but I won't. The tea budding. I showed some tea budding. The tea budding is done in the late summer or early fall, August, September, using current year growth with leaves. And each bud is actually grafted with not the whole leaf, but just the petiole. And you sort of stick it in there and it's alive and you wrap it up. And if it takes, the petiole senesces normally, right? And then the following spring, that bud grows. If the petiole doesn't senesce, if it just dies on there, it's because the bud died, you killed it. And tea budding works really well for like peach trees, and you know, that's how they grew peaches and pears, and apples can be tea budded. Chestnut is a low take. But you can graft on the first year seedlings. In other words, those seedlings could be 0 years old. So I plant the seed, they grow up for, June, July, and August. I T-bud them, even if I only get 50% of them to take, the next spring, they're leafing out. I'm ready to do my whip and tongue graft. If half of them are already grafted with T-budding, and T-budding is really quick, you can do a lot. You can do 100, anybody could do 100 in a day. I've only got to do now, half of them need to be grafted again. So I hope I answered your question. There are lots of the timing. The grafting method that I prefer uses a dormant scion stored until the stalk is active. And that could be April, May, June. Now I've grafted as late as end of June, early July. The problem there is that you don't get enough new growth to really harden off the plant before fall.
Hill, I've got a quick question, and then I'm going to turn Q&A over to Hoy. Joe Kirk asks, I plan to graft transgenic chestnuts onto wild-type American chestnuts. And so I think the same question could apply to if you put any, say, disease-resistant scion onto a susceptible rootstock. What happened? I've had several people ask me this question sort of in the reverse. If I put an American chestnut scion onto a Chinese chestnut rootstock, will the disease resistance be transferred into the scion?
No.
That's not.
And so maybe I didn't understand the question, but let's start over again. Each part maintains its own complete genetic integrity. So if it's susceptible, it's susceptible. In fact, that's why the germplasm conservation orchards will fail. They will get blight. They will die. It doesn't matter what you grafted onto. So I use Chinese rootstocks only because they're resistant to Phytophthora and chestnut blight. And I know that the blight's not going to kill my stock, but the graft union itself, the where the wound is, the graft union is a blight magnet. If there's a blight spore in the stratosphere blowing by, it's like attracted to the graft union. Okay, and so the transgenic tree is not going to transmit any mystical particles to the rootstock. The rootstock will be blight susceptible forever. The scion will be blight resistant until the rootstock gets blight and dies, and then the scion will die. If you could grow it on its own roots, if you could take a cutting and root it, then you've got the whole plant is one thing. Did I explain it? Did I answer that question?
I think you did. So one real quick question. Hill, do you have I know you said you're going to work on this sort of more methodological, procedural, like workshop video. That'll probably be a much longer thing, right? Would it go into more detail? You don't know yet. Okay.
I'm going to have Brandon, who's my intern, stand behind me with his camera and film me grafting. And so while I'm grafting, I'll be talking about it. And I'll explain I'll sharpen my knife and I'll do the cut and I'll explain what I'm doing while I'm doing it.
All right, good. Well, we'll look forward to that. I'm going to turn the Q&A over to Hoy. Hill, thank you so much for coming. It was good to see you, and I'll check you guys out later.
All right. Thanks, Sarah. So a few other questions that we have. Hill, one of which from Ken Law, has anyone had any experience with green on green grafting?
Well, sure. I showed an example of green on green, which is the whistle graft. You do that in June. And so you go out and you collect. So the tree grows a flush and it's not all the way woody yet, but at the base of that shoot, there are nodes with leaves. You cut off one of those nodes and it has to be with the bark is slipping. I'm doing like this because that's how you slip the bark off. And then you've got the shoot over here of a stalk. You make the same cut, you slip that ring of bark off, and then you put it's not really the bark, but it's the rind, all of those phloem tissues, and then you slip that over the end. Now, that's green wood. It's current year, and you would do that in June. That's an example in chestnut. you don't want it to wilt. If it's got leaves on it and they wilt, you're doomed. It's not gonna grow. And so for that whistle, you've got one bud, but you cut the leaf off, and you're gonna hope there's an axillary bud. There's an axillary bud. Here's the stem, there's the leaf, right? You're gonna axillary bud. You got to make sure that bud has to shoot that current year. In other words, you're hoping for a second flush. I hope I answered the question.
I think that answers
Yeah.
it.
So we've done a similar, well, I don't know if it's similar, but really small. Laura Georgie published an article a few years ago on a technique that Fred Hubbard really liked at Meadowview, which was epicaudal grafting, where instead of growing a rootstock, you just grafted it directly onto a seed, essentially. So he had the seeds germinated in a bucket with some sawdust, and we just took the seeds out, and they have a root going down. There's the radical going down. They have a little plumule coming up, and right there where the cotyledons join the stem, we snipped them off right there. We left the nut on there, and we just jammed the scion wood. We just jammed the scion right down in there, and it was really small. They were dormant scions though. They weren't really green. But I'm thinking that if you had some current season shoots that were hardened off, they weren't going to wilt, that technique might work. You could try that. I've not done that, but you could try that. Anyway, check out Laura Georgie's article. It was published a few years ago in the journal.
All right. We've got a few folks sending in alternatives for glue or grafting wax. I don't have any experience with these, but perhaps you do. One is lax balsam, a clear silicone caulking, or Flex Seal. Do you have any experience with any of those? And if so, how do they work?
I like Trowbridge's grafting wax. It's a beeswax lanolin mixture, and it melts at human body temperature. It is, in my humble opinion, the ideal grafting paste. Okay. I don't have any experience using the silicone based. What you want is something that's airtight, waterproof, and infinitely expandable. In other words, the tree's just going to grow through that, and it's a perfectly good material. A box like this, that could last 20 years. You use just enough to cover the tip of the scion. You get a box that size, splurge. But that's a lifetime supply, unless somebody steals your grafting kit, and then you have to buy another box. There are all sorts of grafting paints and tree coats and stuff like this that are not ideal because, for instance, latex paint, for instance, creates a barrier. It's elastic, and you might think it works good, but it creates a barrier that moisture gets underneath it because it's not waxy, it's not sticky, so moisture gets underneath it, and that provokes fungus growth. And so these latex paint-based tree wound dressings, we know now, are actually very contraindicated. You should not use a latex paint-based tree wound dressing. This is bad. It accelerates the fungal decay.
All right. Just a few more questions. Is it possible to successfully air layer Castanea dentata American chestnut?
I've never seen any reports of it. I know that people have tried. I've never seen it. Chestnuts in general are difficult to root, and they vary in across the species. There may be some. I'm sure there's some genetic determinant. In my own experience, I tried rooting cultivars and species chestnut. I tried for many years, and the ones that I got to root were the Euro-Japanese hybrids, the European times Japanese hybrids. I was able to root them occasionally, low rate, but I got them to root. The European varieties, I was never able to root. American chestnut, I've never been able to root. Now, different people have different experiences. I was trying to get roots on mature I was trying to propagate a mature bearing tree. If you take a cutting apparently from a seedling, and this can be done, if you have a seed that's coming up and you cut off the top, and they'll root. They root readily, and lots of people have done that. But air layering, I've never seen adventitious roots form off the trunk of an American chestnut. I've seen mud packing, years and years of mud packing, right? I've never seen a new root form in a mud pack.
Okay, looks like just a couple more questions.
Yeah.
What is your percent take with chip budding and the viable technique?
So I prefer the whip and tongue graft because I think I get a higher percentage of take. I've done a lot of chip budding because it's really quick, and the advantage there is if I had really unlimited number of rootstocks and good scion I can chip bud a lot faster than I can do any other kind of graft. It's really quick. And the other thing about chip budding is you could put 3 buds on one stalk, doink, 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 and one of them will grow, right? So maybe. If I get half of my grafts to live, I'm really happy. If I get 50% success, I'm like that's good. They can die for all kinds of reasons, and they can die because the graft doesn't form, the failure of the graft union. They can fail a year later because you forgot to water them 2 days in a row and they croak. They can get blight and die a year later. And then we have this phenomenon of delayed graft failure, which apparently, and I'm thinking is probably some pathogen. It could be a virus, it could be a fungus, it could be some endophytic fungi, but it's obvious. It's a stress-related response where the tree shuts off the graft union. It just shuts it off, and you get a necrotic layer that forms right at the graft union. And this could be on a mature tree, 10 years old. And in response to this stress and maybe some pathogen, they just shut down. So anyway, I know I'm answering the question about chip budding. I don't do much chip budding, and I haven't effectively done chip budding in several years. I'm just doing mostly whip and tongue and bark flap.
Okay.
The bark flap is really easy, by the way.
Yeah, very easy and fairly forgiving with different size.
So the problem with the bark flap is that it leaves a larger wound. And so if you cut the top off and you put your little scion over here, you've got this exposed wood here. And if you don't protect that from blight, it's going to get blight right there.
Okay. A couple other questions is, how long does a graft joint need to be sealed with wax?
Forever.
Forever.
I don't go back and repaint it after it's growing, but I'm going to look carefully if there's still exposed wood, it's at a risk of blight infection, right? And so at that point, I might use some fungicidal paste. I've got a product that I smuggled home from Italy. It's a tree wound dressing that has a systemic fungicide in it. It's not labeled for use in the United States, and so it's illegal. So I'm not going to tell you what it is because I would get in trouble. But so I've used that before, and I didn't do the whole fungicide slides, but we're sort of in a new era of fungicides now. Steve Jacobs at Clemson University has been bugging me for years to do an actual trial on fungicides. We've got better fungicides now. We have truly systemic fungicides. It's probably time to do we should do some experiments like deliberately on chestnut grafts to see if we can protect them from blight infection using fungicides. I don't know if I answered that question or not. Well, I bungled that one.
No, I think you answered it with. Yep.
The wax keeps it from drying out until the union is formed. And when the tree's growing, if you've reestablished the vascular continuity, you don't need the wax anymore, right? At that point, you're protecting it from something else. After it's growing, you've got to protect it from blight. So in Europe, I showed effectively the use of hypovirulent strains as a biocontrol, where the farmers, these are growers, commercial chestnut growers, will inoculate their trees with the hypovirulent strains. In fact, they had a campaign, organized by the local extension agents where they taught them how to do it. They came in, they did the type matching, they isolate locally to get the type, the VC, the vegetative incompatibility type. Then they took them back to the lab, they convert them to hypovirulent, and they bring you back your own strain. Don't buy hypovirulence on the internet. You want to have your own strains, your local strains converted. And they did that very effectively in France, Italy, Spain, Portugal. They're using hypovirulence as a biocontrol very effectively.
All right. And it looks like the last question is, you mentioned you're working on a video that you'll post at a later date for grafting, but are there any other videos on YouTube for more information that you would recommend?
I can't recommend one. I had a list of some, of suppliers for I put on the PowerPoint there, a list of suppliers for knives and tape and stuff like that. About a year ago or two, I was looking at YouTube, and I saw a video of a guy doing whip and tongue grafts sitting at a bench. It was apple though, and apple is easy, right? But he had the rootstocks were cut into pieces, so it was just basically a piece of root, and he had the cyanwoods already cut one bud long, and somebody was doing that for him, and he was just making the cuts and sticking them together. It looked like the movie had been fast forwarded. The guy could do 100 in an hour. I mean, easy, right? I mean, it was phenomenal. You couldn't see his hands move. And the comment was, it's got to be fast forwarded, but no, the guy's a monster. And which brings up another thing. Can't you do it with the machine? Yeah, there are grafting machines. You do it. The problem with the grafting machine is keeping the blade sharp. And the way the grafting machines work, they tend to crush the stems. And for apple or pear, it's okay because they callus, and the callus tissue is a sort of undifferentiated tissue like a meristem, and you can get vascular continuity reformed from the callus. Chestnut doesn't callus very well like that. And so you really need, the knife needs to be really sharp so that it slices through the cells and doesn't crush the cells. And that's why the take rate is going to be much lower on a grafting machine, I think, because it's hard to keep the blade sharp, and because of the way they work, the blade just comes straight down. It just sort of chops, which tends to crush. Whereas with the blade, I'm going to slice through. I'm going to use the whole length. I'm going to use the whole length of the blade to slice through the cyan. I'm not using it in a chopping. Does that make sense? I'm not chopping it. I'm slicing it. Did I answer that question?
Yes.
I forgot what the question was.
Had a couple more last minute ones.
Yeah.
You mentioned using Chinese chestnut as the rootstock for American chestnut.
Yes.
Is that combo viable long term?
Yes, it is. I've got trees I have trees, I can show you pictures in the orchard. They're going on 20 years old. They bear fruit. The American part eventually gets blight, but if you're lucky, we've got hybrids. I've got Chestnut Foundation F2s, B2 F2s, B3s that are going on 15 years old, they bear fruit. They grow normally. I've also had trees that were 10 years old and looked beautiful. I've got a picture. I could share my slide. I could go back and share that slide. 10-year-old grafted B3F2 that decided to give up the ghost. If I put this, I'm gonna do it. Sarah's not here to yell at me.
Oh, but I am. All right, we got to wrap this up soon, Hill. Make it quick.
So this is SA333 on Chinese, and SA333 is actually a B2F2 from Fred Hubbard, and these trees are the same age, grown on the same rootstock, side by side in the same orchard. And we noticed one afternoon that the one on the left looked a little bit weird. And at the base, you can see I've got a photograph of the base of the tree. Maybe it's not very evident to you because you haven't looked at a lot of these, but the scion is overgrowing the stock. And we eventually harvested this and cut it, and there's a necrotic lesion right here at the graft union. That tree was dead about 6 months after this photograph was taken. That's an example of delayed graft failure. And it's just one of those inexplicable things. But it got that tree and not this one. They're both on Chinese. So I think I answered that question.
Yep.
Stop sharing.
All right. Well, I see there are no more questions, so I think at this point, I'll turn it over to Lisa Thompson. On behalf of Hill and I, just thanks for tuning in and watching, and I hope you got something useful out of it.
Thank you.
Well, thank you, Hill and Hoy. Great presentations, lively audience full of questions. Sounds like they're still hungry to learn more. I just typed in an answer to a poor fellow who's struggling with grafting. And so I suggested he start with Sarah because I could remember her email address, sarah@acf.org. And then she could get in touch with whoever can maybe help him. But quickly, I just want to let you know, we have 3 scheduled Chestnut Chats coming up on June 12th, same time, Friday at 11:30. Tom Sayeli is going to lead us in how to find and identify wild American chestnuts. On June 26th, the presenters are to be determined, but we're going to talk about American chestnut reintroduction. When are we going to get this into the forest, and how is that going to happen, and what's that going to look like? And then on July 10th, Linda Poland and Andy Newhouse are going to talk about a chestnut pollination workshop. They're going to talk about how to pollinate. So that's going to be all really cool. And we also may have some science lectures on all kinds of different subjects. So just stay tuned to our social media and our emails, and we'll keep you posted on what's coming up. Again, Hill and Hoy, thank you so much. Sarah's off, but she does a great job facilitating these. And you all have a great weekend. Okay. Thanks again.
Thanks.
Bye.
Bye.
Bye-bye.
Bye.